Thank you, Sunil. A very good morning to all the delegates to this welcome. A very good morning to all the delegates and the participants and all the dignitaries who have attended this eSTTP program today. I welcome you on behalf of Seven Hills College of Pharmacy for day 2 of online STTP on molecular neurosciences. I hope today we are going to have another informative session by Dr. Ravikumar, sir, Chief Scientific Officer, Immunocure Discovery Solutions, Hyderabad, on the topic of drug discovery in the age of AI, advances and limitations. On behalf of Seven Hills College of Pharmacy, I extend a warm welcome to you, sir.
Thank you very much. Yeah.
Thank you very much, sir. May I now request Dr. Y. Lavanya ma'am to brief about today's resource person.
Yeah, thank you, Lavanya. Yes, I feel much privileged to introduce our today's session 1 speaker, Dr. Ravikumar Muttineni, sir. We can call him as a rational drug hunter, having 19 plus years of experience in drug discovery. He has done his PhD in computational chemistry. He worked in GVK Bio for 4.5 years and Schrodinger for a decade. He started a consultancy service to serve various organizations, those who are in need of expertise skill in drug designing. During his 19 years of experience, he traveled to almost all the drug discovery research departments in the subcontinent and worked with them, where he learned insights into how and where drug discovery is succeeding and failing and why. And in the last couple of years, after leaving Schrodinger, Sir has started developing processes and models to overcome the hurdles in drug discovery research and working presently as Chief Scientific Officer for Immunocure Discovery. And currently, Sir is collaborating with various clients to design and discover new small molecules as a drug candidates using computational and artificial intelligence tools. Sir has involved in developing of customized artificial intelligence models with the in-house curated disease and target small molecule databases to understand the pharmacophore patterns, generating the focused chemical space by coupling with the computational methods to identify the promising leads. Sir has his own preclinical ADME toxicity models, which helps in identifying the druggable compounds. And Sir has published more than 70 research articles in peer-reviewed journals, and to his credit, he filed 10 patents in India. Sir is having extensive experience in biologic modeling, including antibody modelings and vaccine designs. Sir has expertise as well in formulation optimization using atomistic and mesoscale simulations to understand the miscibility and stability of the compounds in order to identify the impurities present in it. And Sir has a very extensive experience in machine learning and deep learning methods for de novo library design. And Sir is technically expertised in computational method development in drug discovery, including binding site analysis, protein modeling, docking methods, scaffold hopping, and fragment based drug design, pharmacophore modeling, molecular dynamics, water thermodynamics, free energy perturbations, quantum mechanical studies, and computational peptide design. Sir also having technical expertise in application of development and data automation using Python and development of GUI to display the studies. I am very much privileged to introduce our today's speaker for the participants who can look back into their research for their betterment. Thank you so much, sir. Now the virtual stage is yours, and please go on, sir.
Thank you, Dr. Lavanya, for the nice introduction. And also, I'm very much thankful to Seven Hills College and the principal for inviting me for this presentation. Uh, and good morning to everybody. Yeah. So I will start my presentation. I think you can see my screen, I hope. And before that, I mean, how is my audio? Is it audible to you, everybody? Right?
Sir, everything is fine, sir.
Fine. Then good. Yeah. Now, right. Yeah. So we are now in the age of artificial intelligence and machine learning. Uh, and So now these machine learning methods, which compiled in artificial intelligence, how these methods can integrate in the drug discovery, okay, where we can we can decrease the attrition rates, which are very important in the drug discovery, which can we can reduce these attritions with the help of these machine learning methods, such that okay we can accelerate the discovery with the cost effective and in a less time. These are the two things which are very important in the drug discovery, which is a very long process. Now for the audience now in this okay presentation, just I will put it in very simple, and I try to introduce some of the maybe concepts or the terminology of this machine learning methods, simple to little advanced methods. And also one of our case study what we have successful, which is into the preclinical studies, that I'll just give a small glimpse of it as such. And but yeah, this machine learning etc is not in a is learning these things in, it's not something like it's a 1.5 hour session, okay, but this will introduce you that okay how these machine learning methods helps in pipelining your discovery process as such, right? So the summary, something like just I want to introduce the company, what we do, and what are these machine learning introductions, what are the methods in the machine learning and deep learning. These are not something like in detail, it's a very simple way, okay, very short, okay. uh, and So how we come up with the broad spectrum anti-cancer compounds by using these methods, okay. So these are the things which I will discuss, okay, in the course of maybe 1.5 hour session, okay. And maybe I will try to complete in maybe 10 minutes before, such that we can have a discussion or maybe a Q&A, question and answers, okay, right. So first, what does Immunocure do? Okay, this is we I work in various companies and then travel to various R&Ds in India and abroad. Then I came to know that there are some lacunae in the drug discovery. So to overcome this, okay, so I thought to start a new company where which can reduce these, voids or lacuna and accelerate the drug discovery. That's what the main aim in establishing this company, okay, because I'm closely working almost 19 years in the R&D session, right? So Iminature is a CRO company, contract research organization, discovering the small molecules. Now we are mainly contained to the small molecule drug discovery, and also we are also involved in the innovating the new formulations, okay, by using these cutting edge technologies like artificial intelligence and CAD, computer aided drug designing, for our clients, such that it will accelerate the discovery, and also the cost of this discovery will be highly reduced by using these methods, okay. How and why, I'll just let you know in the course of our slides. And it is very 1 year old company, so it's established in 2019 October, okay. And as present we have a facility in the cloud. We use AWS for AI, and computational chemistry we have a servers uh, where we do these simulations etc. And we have a medchem facility. So we do the after generating the new ideas, new molecules, we synthesize the compounds and then we supply those things. to the clients. Okay, so and this is, I mean, in it's headquartered in US in New York, and the workspace or the back office, everything is in India, Hyderabad. It is in IKP, Genome Valley. And yeah, this is what the figure when you go to our site. Okay, you'll see this. Okay, so we use AI and computational methods to generate the ideas or hypothesis. Okay, and using this hypothesis, we try to design or predict the new compounds. Okay, and designing and prediction of that. Okay, if you get a very good, reasonable, good compound, then we do the synthesis. Okay, and then we have a tie-up with the biology company, and we test those compounds in the biology, how much they are getting activity or not. Okay, that's how we do it. Okay, so rather than the chemist who synthesize n number of molecules and come up with the desirable compounds, okay, we straight away come up with the rational compounds which can have a better activity as such. Okay, and so this is what the main core concept of the Immunocure. And with this, okay, because we work with the especially with the atom level. Okay, anything, we are in the chemistry, when you know the concepts of the atom and their behaviors are jigglings and wigglings of the atom with respect to others, and if you can decode that, it can be applicable to even to small molecule drug discovery, formulations, or cosmetics designing, and even in the agrochemical discovery. That's what our main of serving catalog in different CRO companies as such. So why Immunocure? As I told you, we are rational drug discovery company where we generate the hypothesis by using the AI and computational methods. For that, we need to have because when you want to generate a hypothesis, okay, so based upon the known information, okay, we try to generate the hypothesis. Okay, so for that, we have a huge collection of known compounds of different diseases. Okay, and also different formulations. Okay, and in the agrochemicals, and we put it into a repository of database. Now we use that and generate the hypothesis by using the machine learning methods, or maybe using the cat. By using this, we try to generate the hypothesis or the knowledge. And uh, and apart from this, when you are working with the drug discovery, the molecule whatever you design in the computer, it should be active against the human, and not only that, it should be having proper druggable properties. Okay, so for that we have a separate engine called as ADMET engine. Okay, so we use all these in combination, okay, in silico, in computer. Okay, and then we use these and generate the new molecules. Okay, we generate the new molecules and then test those molecules in silico with with the help of this hypothesis. If they are passing through this hypothesis, then we do the testing, synthesis and testing, such that thousands of molecules can be reduced to few molecules and have a good activity. As it is a data driven method, okay, definitely it is a disruptive technology, and especially in reducing the time and in the cost. Okay, so this is what we do with the Immunocure. Okay, and so to do this data driven method in the drug discovery, okay, in the drug discovery pipeline, so we have different databases or data sources we obtained from the commercial and also from the literature, and generated these hypotheses. Okay, especially right from the target validation to the clinical, we have different databases. From there we generated our own engine as such, so which can I mean, cater the artificial intelligence pipeline throughout the drug discovery process. Okay, and accelerates its process at each and every step. Okay, and before that, okay, if the people are new to the drug discovery, okay, just give a few important things, okay, the total pipeline, okay, how the drug discovery happens as such, okay. So something like, right from the target selection and validation, okay, selection and validation, this target selection and validation by using the comparative genomics and also by using the bioinformatics, okay, they try to identify the culprit target, okay. Culprit target means the disease causing target, okay, and then based on that target, okay, they try to identify the molecules which can bind to the target, and then try to inhibit or maybe activate the target to decrease the disease condition as such. So initially, when they start with the discovery, okay, the people says that hit identification. Okay, hit means at least whether it is binding to the target and showing some activity. That we'll call it as hit. Okay, and then once you get the hit, okay, then what the people will do, whether if you modify the groups, okay, then whether it is changing the effect or based on the changes in the substitutions, okay, the activity of the protein is going to be varied. Okay, so to understand, we can call it as a SCR, structure activity relationship. They try to understand, and not only that, whether the molecule can be druggable, okay, that they try to identify in in hit to lead option. And then from when you get a lead molecule, okay, this target can be druggable, you can target that. And if you can if you modify the molecule, then it can change the activity of the target. If you get the establishment of that, okay, then we do the lead optimization. Okay, in the lead optimization is nothing but multi-parameter optimization. I will just tell you what are those things in our slides. Okay, we have a beautiful slide where we do that. Okay, we'll go through that. So multi-parameter optimization means getting high activity and good druggable properties. We'll call it as a multi-parameter optimization. We do it. So, and once you get a good activity, okay, and desirable druggable properties, then we'll test those molecules in the preclinical, maybe in the animal models, okay, maybe non-GLP to GLP, we do that, and then generate the data, okay, and then we do the IND enabling studies as such. Okay, this is what the discovery part of the drug discovery cycle. And then once IND file, that molecules enters into the clinical stage, clinic. So it has phase 1, phase 2, phase 3 clinics, and that is a time consuming. It's almost 10 to 15 years of 10 to 12 years of time in the clinical trials. Maybe some molecule drugs, okay, which are rare diseases, okay, sometimes they give a accelerated FD, clinical trials, where you can complete it in 5 to 6 years. Okay, but the discovery part, at least it takes to, maybe we can say that 3 to 5 years of time. And in this, lot of molecules are synthesized, tested, and lot of animals will be, gone through these studies, and you need to sacrifice many animals during this study. Okay, so to overcome all these things, okay, this artificial intelligence or maybe machine learning computational method will reduce the number of molecules and reduce the cost by reducing the synthesis, and also by reducing the biology as such, maybe reducing the animal killing and all those things. Okay, so this is what the steps involved in the drug discovery. To be just a recap, one is target identification validation, so hit identification, hit to lead optimization, preclinical, and IND enabling and filing as such. Okay, and then clinic. These are the things. Okay, now we are going to use our methods, artificial intelligence methods. Okay, to address these, the steps in the drug discovery as such. Okay, right. Remember this. Okay, so now, yeah, as you see that the drug discovery is something like, it's not that simple process. Okay, to identify a drug molecule, when you put together, this is one slide I always show it to the people how complex the drug discovery is. Okay, if you want to synthesize a molecule, to a particular target and should be druggable, for example. Okay, then how you can start? Okay, the reason is there are, if you put together all the synthesizable compounds, whatever there in the world, okay, if you put together, okay, all the synthesizable data, okay, it is something like 10 to the power of 60, something huge number. Okay, in this huge number of molecules, okay, few of these molecules are active against that particular target, culprit target. Okay, among these active compounds, okay, only few molecules becomes drugs because they have desirable druggable properties. Okay, so now what is the thing is, in the huge chemical space, we are cutting down to a small number which are actives, and from there we are cutting down to the, very small units, okay, which can be a drugs. Okay, so this, if you want to implement, it is a highly time consuming. That's why, drug discovery is not that easy to come up with these molecules in this particular huge chemical space, latent space. Okay, so that's the reason. Okay, okay, people use this, the um, computational methods to reduce the time as such. Okay, so, and now, okay, another important thing, okay, is the drug discovery is not that, we started now or maybe a decade back. It's a continuous process. Okay, from very old, okay, to now. Okay, drug discovery is being a continuous process, and so we are going to get the hit molecule for the target or maybe for the disease. Okay, and get the molecule and we document that, and then see whether it's a drug, how it is working on the animals. All these experiments we do it in the course of, maybe in the advent of new, sophisticated techniques and equipment, parallel synthesis, screening, etc. Okay, then this large experimental data have been piled up, and well, and are maybe sometimes organized in some cases and may not be so organized in other cases. Okay, but the data is available. Okay, recently several, you can say that data, you can also go through them as such. There are several chemical databases, okay, which are available in the internet, like you can see here, like PubChem, okay, and drug bank, remember zinc, okay, binding db. Just why I'm just reading out, because these databases contains the information of the molecules and their activity against different diseases and other clinical aspects of it. Okay, when you put together all the compounds from these database, it's approximately maybe 100 million compounds available, okay, in the form of databases, etc. And now, okay, and now, sorry, Uh, just in between, I'm just interrupting you. I'm audible. Everything is fine now, right?
Hello,
Just
sir.
small
It's audible,
interruptions.
sir. It's
Yeah,
audible,
yeah.
sir.
So
Thank you, sir.
please, disturb me if I am not audible. Okay. So
Sure, sir.
please,
Thank
okay,
you.
because it's the internet, we don't know. Okay,
Yeah,
I'm
sir.
not seeing your faces as such. Just disturb me as such. Okay,
Okay.
right. Thank you. And
Thank you, sir.
yeah, so just pardon for the, small break. Okay, now we have something like 100, we can say that, 100 million compounds with activities against various diseases, and there are drugs, okay, and their clinical data, and also ADMAT profile data like solubility, Kakosu permeability, like liver toxicity, that's hepatotoxicity, okay, cardiotoxicity, etc. All the data is piled up. We can put together it have with something like, for at least we have 100 million compounds with the different data sets. Okay, now it is known, actually the people have generated the data by doing the experiments. And not only that, okay, we also know several biological targets. I told you, what is a target? It's a therapeutic drug action generally occurs by changing the activity of the functional protein that we'll call it as a target. It might be protein, DNA, RNA. Okay, so when you put together, there are something like 3,419. Okay, we have already for drugs which are there in the market, there are something like 461 targets which are there. Okay, targeting those targets, and clinical targets maybe 1,000 plus, and patented target 201, research targets, okay, something like 1,500. Okay, and accordingly we have, approved drugs are 2,600, and clinical trial data something like 9,000, just around 10,000, and patented compounds are 5,000, and experimental 20,000. Like that, we have the biological targets and the known data information. Okay, and there is a beautiful site. Okay, every pharmacy guy or maybe the people who are into the drug discovery has to visit this site called as Guide to Pharmacology, which has the information for each approved drug and also clinical molecule. Okay, and the information available in a very very queryable format. Okay, in this guide. Okay, and it was just done by the International Union of Basic and Clinical Pharmacology and British Pharmacology. Actually, these people have generated this data. It can be a something like, very good pharmacology book for the people who are going through the, uh, pharmacology. Okay, in a very queryable format as such. Okay, and Campbell, Drug Central, Cancer, these are all the databases with small molecules and known targets as such. Okay, so if this data is available, if you put it in a proper organized way, and if you use this knowledge and design the new molecule based on this knowledge, then you can be more successful. That's what the idea is. When you have a known things, okay, known information, if you use it, then it can be useful for, new molecule designing. Okay, that's the reason. Okay, James Way Black, okay, is a Nobel laureate, got Nobel Prize in 1988, especially for the working for propanol and cimetidine. Okay, propanol is antihypertensive, both are antihypertensive drugs. Okay, these sorry, cimetidine is the histamine blocker. So he got the Nobel Prize, okay, in 1988. Okay, and so getting one drug molecule is a difficult task in the lifetime. Okay, he could able to generate two different drugs, okay, which is nowhere related. So that's why he was awarded with the Nobel Prize. And what he told, famously told, the best way to discover a new drug is to start with an old one. Okay, this is what a very valuable information, where if you have a known things, okay, information, if you put together and use that knowledge, then you can come up with a drug in a very less time. That's what this, completely our machine learning or computational methods work on it as such. Okay, so based on this known information, okay, we will predict the new molecules or new drugs. Okay, and this we'll call it as this, machine learning, artificial intelligence, deep learning, whatever you say that. Okay, we call put together as a predictive modeling. Okay, what is the predictive modeling is, with the help of the known information. Okay, we use mathematical process. Okay, that seeks to predict the future events. It's going to predict the future events or outcomes by analyzing the patterns. Okay, what patterns? By analyzing the patterns of the known compounds for a particular disease, for example. Okay, which patterns make that molecule active against that particular disease. Okay, and based on that, okay, analyzing the known patterns and identify the patterns which can forecast the future. Among these patterns, solubility is very important for the biological activity, or maybe you can say log P is important for the biological activity. Okay, now for the known active compounds, using the mathematical process, it tries to predict the new future events or new drugs by analyzing the patterns of the existing molecules. Okay, and understanding the forecast futures of the results. Okay, so this we'll call it as a predictive modeling, and this is a funda for any kind of artificial. It might be in the drug discovery or in the formulation or maybe in other field, okay, mechanical engineering or whatever it might be, okay. So the funda is take the known information, that is a known data, and see the analyze the pattern, see which patterns will give the future or results, okay. That try to understand with the help of the mathematical process, and using that you try to design the new molecules, okay, new events, okay. This is what the predictive modeling, okay. And this mathematical process, all these things, algorithms, we put together as a machine learning. And now if you make a, for example, all the algorithms, if you put it in an automated way and make it as a engine, complete engine from end to end, then we'll call it as artificial intelligence. Artificial intelligence is an end-to-end process, something how human will predict, okay. So it also predict without much human intervention. It predict the total process, okay. For that we have a, it should have a something inbuilt intelligent programs, okay. Or we can call it as a inbuilt machine learning methods, okay, in an automated way to solve the problems. So all you put together as artificial intelligence, and in that artificial intelligence the subset will be the machine learning as such, okay. And in the subset will be the machine learning, this algorithms, everything we call it as a machine learning, which has something automated process, okay. And these machine learning methods will have these mathematical process, okay, different mathematical process to predict the new things. That's what it will do it in the machine learning. And in that, okay, there is another subset called as deep learning. Deep learning and machine learning is having a very thin line. We'll discuss it later, okay. Where deep learning is one of the machine learning methods, where it has a something like a neural network, a complex neural network which is there in your body to function of the human body. You have a complex neural network to understand the complex patterns, because molecules, the ADMET properties, etc., they're all complex issues. So to understand the complex patterns in the molecule against a particular biological response, okay, then we generally use a complex neural network, and that neural network will call as a deep learning as such, okay. So these are all machine learning and deep learning, all the kind of algorithms are having a integrated machine mathematical process to understand, to analyze the patterns, okay. And when you put it into a end-to-end process, okay, then we'll call it as artificial intelligence as okay. Now, okay, so what is the workflow? Okay, now I just put it as now machine learning or deep learning. I'll just say that whatever the terminology, okay. But in any process, okay, now you understand that it has the mathematical process, okay. So how this workflow will work, okay, is, for example, I told you that these are all predictive modeling, and these are all data driven methods. We need to have the known data, okay. And known data is nothing but the experimental data you collect from the literature or from the different sources, okay. So that will take it, okay. The known data will take it as an input, okay. So for any machine learning method, what is that? The training set, okay, or we can call it as a input data, known data, okay. For example, for a cancer we are working, we collect all the anti-cancer compounds, okay. And those compounds will call it as we'll call it as a known data, and also call it as a training set as such, okay. Because this, using this known data, we try to train and generate a hypothesis by using machine learning methods, okay. That's why we'll call it as a training set as such, okay. And now in this methods, okay, in the training set input data, it can be of machine learning, we can be of two types. I will tell you later, okay, but whatever, supervised and unsupervised, okay. I'll tell you what is supervised and unsupervised, but the input data is required for both the methods. And then it do some pre-processing called as it try to see what are the patterns, what are the features, what are the properties present in it. It try to do the feature extraction, okay, for the input molecules. And then now based on these features, okay, now you try to generate the machine learning models, okay, by using the mathematical process as such. Okay, so what you try to see now, if you have a something like a biological endpoint, because anti-cancer, this is having a high active, low active, moderate active, like that you have it, or if you have a activity of some nanomolar, micromolar, like that. Okay, so for the given molecules, okay, try to see how what kind of patterns which makes the molecule as anti-cancer, that you try to generate the knowledge or the hypothesis in using the machine learning methods based on their features or the patterns. Okay, and based on this it try to generate the predictive model. Now these patterns contribute for the activity, okay, and the activity can be high, moderate, or less. That type of predictive models it try to generate it as such. Okay, now for example, if I give a new molecule, okay, and the new molecules also having new molecules means unknown data, whether it is an anti-cancer or it having activity or not, we don't know. Now we have given that, it try to do the feature extraction using this machine learning method already which is generated. It tried to predict that whether this new molecule is having, okay, uh, anti-cancer activity or not. If it is there, how much activity, it try to predict it, because we have a training model, machine learning model, which says that based on these patterns this is having either anti-cancer or not. If you give new molecules, you try to see the patterns. Based on the patterns, say that it has an anti-cancer or not. Okay. Now, before doing the synthesis and testing, if you haven't designed the new molecule, if it is going to say whether it is active or not, that's good enough. Okay. We'll take only the active molecules and do the synthesis and testing. Okay. This is how this machine learning work in a nutshell as such. Okay. So it should have a, training data, feature extraction, property extraction, and using the algorithms, okay, it generate the model, and then using the predictive models, we can predict the new molecules activity. Okay, sometimes you want to put the molecules into different clusters. Okay, or for example, I want to put the molecules, anti-cancer compounds in one, anti-diabetic in one cluster, like that you want to do that. Okay, I can, using these machine learning methods, we can group the molecules based on their chemical space or disease based spaces as such. Okay, but disease based spaces is a little difficult task. Okay, but however, based on the chemical space, you can group the molecule, means the quinoline compounds in one group. I can say that um, purine compounds in one class, okay, pyrimidines in one class, like that. You can, based on the chemical space, you can group the molecules into, okay, into different clusters as such. Okay, you can do that, or maybe you can generate a predictive models as such. Okay, this is what the machine learning workflow, what it will do. Okay, in a simple way, we can say that we can take the input data, analyze the data, find the patterns, okay, by using the machine learning, and predict them, okay, and use it for new molecules predictions as such. Okay, so if, in the same way, you can see this, if you have a molecule, for example, and this is one, red color is one pattern, and you can see the green colors are some other important patterns. Okay, you try to see the patterns in the molecules. Okay, and we'll, these patterns we call it as a descriptors. Okay, something like hydrogen bonding acceptor 3, hydrogen bond acceptor, this is a donor. Okay, aliphatic chain, like that. It has a different patterns. Now based on this pattern, what is the biological activity, we can know it, because this is the input data. Okay, for the input data we know, for the input data we know the biological endpoint, and we analyze the patterns. And now which patterns makes the molecule active, that we do it with the machine learning. Okay, and if you understand the patterns which makes the molecule active, then now in a new molecule, if you find this similar pattern, then the new molecule will be active. Okay, that's how this machine learnings will try to identify the predict the new molecules. Okay, it get the training from the known compounds, and that applied for the new compounds to identify the activity of the new compounds. Okay, but as this methodology, whatever we are saying, these are all statistical methods. Okay, and predicting the new things. Okay, always will have the errors. Okay, noise and all those things. Okay, so if the model is good, it has a good prediction. If the model is bad, say, doesn't have a good prediction. Okay, then if you synthesize and test it, doesn't show the activity. Okay, so that is the reason. Okay, whenever you generate these machine learning models, okay, they may have some kind of noise to know whether it has a precise method models or not. Okay, so we have some validation protocols. Okay, people use several different validation protocols whether that model is correct or not. After that validation, okay, then they will take this model and predict the new things. Okay, so the thing is, in the machine learning methods, input data, pre-processing, that a future selection, and then model generation. Okay, and then using the model we do the prediction. Before that we do the validation. Okay, this is what the things which we have it in the nutshell. Okay, something you can see this, they will give it in a color way also, the patterns for this molecule. Red will indicate the most active, blue indicates the inactive regions of the molecule, like that is give the patterns as such. Okay, now based on that you can design the new molecules also. Right. Okay, now if you know the patterns and everything, okay, whether this is useful to design the molecules, because designing drugs is harder than rocket science. Okay, yeah, this is very important, because rocket science, we know the most of the times you know the target, and you know the initial velocity, how much you have to give it, and how much I mean the where is the target. If you put everything put together, most of the times, okay, or we can launch a rocket, okay, to the target place. Okay, uh, but in the drug discovery, there are many unknown things in the human body. If you see this, if you take the cell, the complete, cell, understanding the complete cell, okay, is not yet completed as such. Okay, there are many unknown pathways are there, proteins are there. Okay, so that is the reason. Okay, we are playing in the drug discovery, we are playing with the human body, or maybe animals. Okay, where there are many unknown things are there. That's why drug discovery sometimes is a luck, or maybe harder than rocket science, because we need to increase the potency. You should have a high active compound, should be very selective to the particular target, not to the others, and should have a good solubility. Okay, because it should be, most of the time our molecule should be oral, could have a good solubility, and a permeability, should have a good permeability across the cell membranes, good bioavailability. Okay, and optimal CYP inhibition. Okay, that means you should, okay, and clearance of life. Okay, should be optimal. Okay, it should not have high fast clearance or low clearance rate. Okay, uh, and health journal, because this is one important thing for the cardiotoxicity. Okay, and and the other toxins, like organ toxicity, liver toxicity, kidney toxicity, etc. Okay, so all the toxicity should be considered, and should have a good synthesizability. Okay, and stability. All these things which are, we are going to have it in a single molecule. Okay, all these things should be in a single molecules. Okay, from a large library, all the filters we have to go through it and come up with a drug molecule. Okay, that's why this process is called as a multi-parameter optimization. Okay, multi-parameter optimization, from not only the activity and ADMET. Okay, we need to properly optimize it as such. Okay, so that's something like PK and PD, both we need to optimize. Okay, from a huge number of molecules we need to cut down to the less molecules. Most of the companies, okay, big pharma, or maybe the more medium sized pharma R&Ds, okay, they have a proper pipeline. Okay, where from the initial stage onwards they try to see the molecule whether they are having any kind of efficacy, selectivity, and also the early identification of ADMET properties. They do it in the early drug discovery techniques as such. Okay, they use a lot of computational and machine learning methods to do early identification of druggable candidates. Okay, they see that, okay, they calculate the activity and see that molecules which are having, okay, high efficacy. Okay, and you can see that red colors are not having high. I mean, you can say that, unfavorable, red or magenta unfavorable, only the blue, we can say that cyan are the desirable, white are borderline, and red are, we can say, magenta are not desirable because of the efficacy or other properties like that. They make it in a proper, tabular format and identify the best molecules, druggable compounds, and then go for the proceed for the next level. Okay, but most of the academics in India, most of the times the people see, try to synthesize some compounds and see that that molecule is having activity or not, irrespective of other properties as such. Okay, so even in the academic, in the lab, if you have a proper outlook both on the ADMET and also the other properties, okay, then in a academic lab you can synthesize a druggable molecule, and you can take that molecules into the clinical stage as such. Okay, because synthesis effort for any compound is almost same. So if you come up with the right molecule, okay, in synthesizing the compound, definitely you can end up with the active compounds and druggable compounds. Okay, so this pipeline PK, and not only that, they have the in silico methods for the dosage predictions. Okay, all these pipeline they will use it in the industry. Okay, and all this in silico pipeline will be complemented with the experimental pipeline, and then they try to come up with the molecules, especially from the discovery to the clinicals as such. Okay, so they have complete integrated process of doing it. Okay, so now I just remember this, I just told you something like, what is the machine learning, and how, what are the mean application, means how we can use it in different areas as such. Now we'll see that what are the methods which are there in the machine learning, people use it. Okay, so simple methods to little complex methods. Okay, and then we'll go to the our one case study as such. Okay, now, yeah, now simple, before that, okay, remember this, these are the two important things we need to have an idea. Maybe you know it, I mean, it's very glad for that. Okay, but if the people doesn't have the knowledge, okay, okay, on the molecule, and the important thing is smiles, simplified molecular input, uh, sorry, input entry systems, smiles, okay, is a 2D representation of the molecule, okay, based upon their I mean connections, okay, type of atoms, okay, and their ring systems. Based on that, they have a 2D representation, and this representations we call it as a smiles, and these representations will take less space. Okay, that's why they, in most of the database, they will provide the molecules in the smiles format, and there are several tools which will convert these smiles to the 2D or to 3D structures as such. Okay, and these smiles notations are very important in the machine learning, because many tools they'll read the smiles, and based on this it will understand the patterns, and then it try to relate these patterns to the druggable properties, or maybe to the active compounds. Okay, so smiles are very important, in our course of slides as such. Another important thing here is molecular fingerprints. Okay, along with the properties, okay, a molecule, okay, based upon the presence of a particular property, okay, like, benzene ring is there, it says one. Okay, uh, hydroxyl group is there, okay, it says one. Okay, carboxyl is there, one. If something amine is not there, okay, then it makes a zero as such. Okay, like the presence and absence of the groups, okay, then it will convert the molecule into a fingerprint, and this fingerprint will be unique to this particular molecule as such. Okay, and based on this fingerprint, okay, which has these groups and their connections. Okay, based on this fingerprint, we can compare one molecule to other molecule. Okay, how much this molecule is similar to other molecule. We can compare one molecule to other molecule. Okay, so now these fingerprints and these smiles are used, okay, very quite oftenly, maybe most of the times, in these machine learning methods to compare the molecules or to design the new molecules. Okay, so that's the reason. Just I want to give small idea about what is smiles and these things. Okay, right, got it. And keep this in mind. This is nowhere related to our artificial, but these the nuts and bolts for our discovery as such. Okay, now we'll come to the types of machine learning methods. Okay, and in this the machine learning methods are broadly, we can say that, classified into supervised and unsupervised machine learning methods. Broadly classified into supervised and unsupervised, because based on their purpose they have classified into supervised and their unsupervised methods. In the supervised methods we have two, once again, regression methods and classifier methods as such. Okay, regression methods and classifier. Most of the times these regression methods, okay, those are all continuous methods, we'll call it as. Okay, and classifier methods are categorical methods, we'll call it as such. Okay, in the regression methods, most popular is linear regression methods. Okay, partial least square regression, PLS methods, we'll call it as. Okay, a Gaussian based process. Principal component means, you know, that principal component analysis. Based on the principal components they do the regression analysis called as a principal component regression methods. Okay, decision tree methods. Okay, like that we are having. And deep neural network, convolution neural network, as you can see that neural networks also implement these regression methods in their framework. Remember, and the important, because just noting down this point, underlying this point, because neural networks is a different kind of methodology framework, but uses these regression methods, or we can say supervised or unsupervised learning methods. Okay, that's what. And regression methods and the classifier methods. Okay, in the classifier methods, these methods will try to classify the molecules, classify the systems. Okay, based on their, you know, nature or inherent property, etc. For example, you want to classify the molecule based upon their chemical, I mean, we can say that, you know, active to the inactive molecules. I have the active molecules and inactive molecules, and I want to classify based on their patterns, actives to the inactives. Then we have these classifier methods, SVMs, or toxic and non-toxic. Okay, so we know the toxicity of all the molecules, and uh, and we know the non-toxic compounds. Okay, then we can use this method to classify the non-toxic and the toxic compounds as such. Okay, classify molecules. And unsupervised methods, where based upon the pattern, the chemical structure, or based upon their inherent properties, it will classify the molecule, cluster the molecules into groups, and those are called as unsupervised learning methods. In this we have very famous k-means clustering, hierarchical clustering, etc. Neural, and you remember this, neural networks and sorry, neural networks will use supervised and unsupervised methods, both the things, in their framework. Okay, to come up with the drug like molecules. Okay, so, and there is another neural network. Remember, this is a gas. Okay, we'll discuss those things one by one in a few in an abstract way. Okay, so now with this slide, okay, what is the thing is, we try to have a supervised and supervised learning methods, supervised regression and classifier methods and clustering methods. What are these, how it will work with, maybe I'll show you in a few slides. Okay, what are the supervised and how they work on it. And so all these methods, okay, uses, okay, large data, process them using the DL and ML methods. By using these methods, okay, it try to generate the knowledge or hypothesis. Okay, that we'll call it as a model. Okay, predictive model. And using this you can predict the new molecules activity. That's what the main essence of this. Okay, what is a supervised learning method? I will just tell you that. Okay, supervised learning methods are used to develop the training models to predict the future values of the data. Categories are continuous variables. Okay, don't worry about this. Okay, simple thing. For example, I know the biological activity of a series of compounds. I know the biological activity of a series of compounds. Okay, means I know the endpoint. Biological activity is the endpoint here. Okay, I know the biological activity of a series of active compounds, and now I see the patterns by using the machine learning methods. Which patterns makes the molecules are active and moderate active or inactive? That I try to generate the the machine learning model based upon the known activity of the molecules. Based on the endpoint, we know that these molecules are active and these are the activities. We know the endpoint of it actually for these molecules because these are the input data. Okay, so based on that, we generate see the which patterns are important for the activity. For that, we generate a machine learning model. Okay, now for a new molecule, okay, if you want to predict the activity, then you see it sees the pattern and then it predict the activity. Okay, based on these patterns, it says this molecule can be this much active. Okay, so if you know the endpoint, based on the endpoint, if you generate a model, and that models are the predictive models, that models are called predictive models. With these predictive models, we can predict the activity of a new molecule. Okay, for example, simple thing, I know that here in a very layman language, okay, I have a series of grapes here. Okay, now I want to in a new cluster of different fruits, I want to pick the grapes only, for example. Okay, I want to generate a machine learning model. So first I will see that what is the grape should have a bunch of round spheres. Okay, and the colors, texture, all these things, patterns, you try to understand in the supervised learning methods. Okay, and then finally, now if you give a new fruit, whether it is a grape or not, it try to identify that. If you give a grape, then based on that machine learning pattern, okay, which is understood by the input known grapes, it says it is a grape or not. Okay, so in the same way, if you give active compounds and inactive compound, based on that it try to generate a patterns, which patterns are favorable for the actives and which patterns are not favorable for the inactive compound. Based on that, it generate a model. If you give a new molecule, based on this pattern comparison, it try to find that new molecule is an active or inactive molecule. Okay, means based upon the given endpoint, okay, it try to generate a relationship, then that is called as a supervised learning method as such. Okay, so in the supervised learning methods are predictive models, and it have known data with endpoints. Based on that, it generate the model. Okay, and most of the supervised learning methods, what we saw, that there are two things. One is um, categorical and also continuous methods. We have it as such, regression methods and categorical methods. And the regression methods, okay, I hope most of the pharmacy and chemistry guys knows about the QSAR, SAR. Okay, quantitative structure activity relations. These are all nothing but the regression methods. Okay, and classification methods available in the QSR. Okay, we are going to see what are the important features responsible for the activity. Okay, so for the known active compounds, what are the features which are responsible for the activity? If you get the relationship, a linear relationship, then we'll get a straight line. Then when you get a straight line, the straight line equation is equal to y is equal to mx plus c or wx plus b, whatever, linear regression line. Okay, then this equation will call it as a QSAR or the mathematical relationship or maybe machine learning model. Okay, so that means for a given set of molecules, based on their properties, chemical properties or patterns, or maybe based on their fingerprint patterns, okay, whatever it might be, what is the how well these properties are correlating with the biological activity, which properties are correlating with the biological activity. Based on that, if you generate a regression model, okay, then the equation is nothing but y is equal to mx plus c, that is called as QSR or a machine learning model. Okay, so if you give a new molecule, then the new molecule's properties can be calculated by using the computer. And if you fit that into the equation, now that property is x, and we know the dy by dx, that is what the coefficient, and b is the y-intercept, we know that. And if you put this equation, okay, for a new molecule, we can predict the new molecule's activity as such. Okay, that's how these supervised learning methods can predict the new molecule's activity. And in the same way, for example, sometimes we know that you don't have the known activity of the molecule, but we know that the molecules are active and inactive. or maybe toxic and non-toxic, or high hepatotoxic, low hepatotoxic, and moderate hepatotoxic. I want to classify them. So I can use different classification methods, okay, and define, and based on the patterns, I can classify why these molecules are toxic and why these molecules are non-toxic as such. Okay, there are tools called as SVM, etc., which uses this method classification as such. Okay, but toxicity, activity, these are all endpoints. Based on these endpoints, we are generating the we are training the model. Okay, that's why we'll call these are all supervised learning methods as such. Okay, so in our, as we uh, told that we are mainly into the drug discovery, we have our own ADMET engine, okay, where we collect the data. We have collected almost 15,000 samples. Okay, it's unpublished data, 15,000 compounds for the solubility, experimental solubility. Now, based on their patterns, okay, we try to generate a solubility model. We took almost, okay, we divided the molecules into training. Okay, we divided the molecule, 15,000 compounds. We divided, maybe we can say 60% of the molecules as a training. Okay, 60% means almost something like, we can say that 10,000 compounds as a training set, and generated a model. In this model, okay, we try to see which patterns makes the molecule more soluble. Based on these patterns, we generated the model, and we got a very good model. Okay, which is something like, good model means it is going to give you a statistics. That's what, because it's a correlation. We'll call it as a good model means it refers to the statistics of that model as such. Okay, most of the important things, statistics, generally people use. There are many things are there. Later we discuss it. One of the important things, correlation coefficient. Okay, when you generate a model and you want to, using that model, you predict the property of a particular model. Okay, using that model. Okay, then the predictions are very close. Okay, then the correlation is very high, which is something like near to 1. If the predictions are bad, okay, means I want to predict the solubility, okay, and the solubility is showing very bad, okay, for by using this model, okay, then the correlation will be something like, correlation coefficient will be near to 0. Okay, so for a good model, okay, for the input data, the correlation coefficient, if it is near to something like, near to 1, then the correlation coefficient is, the model is more accurate, more precise to predict the new molecules solubility. Okay, and I don't know how it predicts, whether it will do it or not, whether it is having good noise, good prediction or not. To do further validation, what I did is, I have 15,000 compounds. 60% I use it for the model generation. Remaining molecules which are not used for the model generation, with the known activities, whatever known solubility, I use it to predict those molecules with the help of this model, whichever I generated. Okay, and that remaining data will call it as test set. And the test set correlation coefficient is something like 0.7, means it is something like 70% accurately predict the solubility of the molecule. Okay, for a large pool of sets. Okay, means when I design the molecules, if I use the model, okay, if I use 10 molecules, 7 molecules definitely will have good solubility as such. Okay, so that's what I generated this model, and more rigorously use this model for our solubility predictions as such. In the same way, there is another important thing is cardiotoxicity. Might be you heard about these potassium channels present on the surface of the on the heart, which will control the flow of potassium, especially for the muscle function of cardiac muscle function. So if a drug is blocking this potassium channel, okay, HER channel, we'll call it as, okay, then what happens? It leads to cardiac arrhythmias. Okay, then it leads to cardiotoxicity. Our molecule should not block it. Okay, so that's the reason I generated a model whether this molecule, whether my molecule can have cardiotoxicity or not. That predictions I did it, okay, by using this model. This model I generated with the 4,000 compounds and got a very reasonable model. Okay, these are all generated by using the regression method as such. Okay, So I got a straight line equation. Again, I use this equation to predict the new molecules property. Okay, so like that I use a Kakoto permeability, etc., those things. Okay, and next one is mutagenicity. Mutagenicity is something like complex term. We don't get the activity like, you know, so this much solubility, this much for each molecule, like that we don't get it. But mutagenicity will give that, you know, mostly we can say that they will give the result in the form of, you know, highly mutagenic, moderately mutagenic, and non-mutagenic, like that they will give it. Okay, or either mutagenic or non-mutagenic. That's what the mutagenicity endpoints will in the published results will have it. Okay, so I just collected 6,000 samples, 6,500 samples. I use a categorical model method like support vector method to classify mutagenic to non-mutagenic as such molecules. Okay, I got something like a very precise results of 80% accuracy. I got it. Okay, so this method model, now you can predict the new molecule whether it is mutagenic or non-mutagenic as such. Okay, so that's how these classical, these supervised methods are useful in the activity endpoints and also the ADMET endpoints also. Okay, and this is another important of machine learning methods. Okay, one unsupervised learning method, where in this method, okay, we don't have any kind of biological endpoint. Okay, and now in this method we try to see based on their patterns. Okay, can we categorize the molecules into different classes, or in the simple way, based on their chemical class, can we categorize it? Okay, it's not have, we are not looking for the biological endpoint or the ADMET or anything, no endpoints. So based on their inherent property, can we classify it or not? For example, you have different fruits. Okay, now when you put grapes in one cluster, apples in one cluster, and etc., like that. Okay, based on their inherent property. Okay, so these are this methodology where it doesn't use any biological endpoint. Okay, in the input data to train the model. Okay, then those are called as unsupervised learning methods as such. And this unsupervised learning method doesn't have any labeled data or the biological endpoint, and based on the chemical space try to cluster the molecules into different groups as such. Okay, and these methods, okay, are something mostly we can call as principal component analysis, clustering. Okay, are available, different clustering methods are available. Okay, we can say that hierarchical clustering or k-means clustering or density, there are several clustering methods are available to categorize the chemical space into different groups as such. Okay, simply clustering, here we can say that, you know, if you have a small set of compounds. Okay, so there is a method called as hierarchical clustering, where it will based on their, you know, patterns or the fingerprints. Okay, it try to see the molecule, how much the molecules are similar to each other. Okay, based on their similarity or based on their dissimilarity, it will group the molecules into different clusters. If you see this molecule is little bit close to this because this have some group which are near, and if you see these molecules very different as such. Okay, like that, based upon their, you know, similarity, it tried to cluster the molecules into groups and try to give the connections, how much they are similar, how much the molecules is similar. Okay, it tried to give it in the hierarchical fashion. Okay, so this method is very useful for a small set of molecules for classification. But when you have a large set of molecules, these methods cannot work because it takes a lot of time to classify based on their hierarchy of the similarity. So there are several other methods called as a DB scan. Okay, density based scanning and k-means clustering. These methods, okay, these are something random based methods, and try to cluster the molecules, okay, in little bit random or maybe some other fashion. Okay, or maybe we can say that, you know, with some intelligent way, where it takes a highly diverse molecules and pull the nearby molecules and generate the cluster. Okay, in that way try to cluster the molecules. And these methods are very useful when you are working with a large database, very fast, and that can be cluster the molecules with a similar structure, shape, or any other property, whatever you are desired would result as such. So for example, in our for one of our research, okay, we cluster the kinase database molecules. Okay, so there are from the journals, patents, etc. We have taken all the molecules, something like 3 lakh compounds. Okay, we use the k-means cluster, not the hierarchical, but just I want to show the different clustering methods. I cluster the molecules by using the k-means clustering, and then I identify that what kind of fragments, what kind of groups which are there in each molecule or in a cluster of molecules to understand the total kinase inhibitor chemical space as such. Okay, so these are the supervised and unsupervised learning methods, and some of the special cases of the supervised and unsupervised learning methods will call it as a artificial neural network or maybe deep neural networks, because these artificial neural networks and the deep learning methods, they are amalgam of both, sometimes that supervised learning and unsupervised learning method. They have a framework of both for a specific function as such. Okay, so that's why they can be classified into both or maybe in between that as such. Okay, so most of the times we'll say that artificial neural network is nothing but here also the process is same. It have a input data, okay, and hidden layers. Okay, it try to process the patterns based on the type of patterns. Okay, and based on the type of output. Okay, it will give the weightage to the each pattern. Okay, based on the type of output you want it will give the weightage to each pattern. It's nothing but this is input and the patterns, and they are connected as such. Okay, and it's nothing but in this neural network, it has a stack of information, and this information will be connected to the input. To the input is the input data, to the output result, desired result, and these connections are based upon the output. The weightage of these connections will be based upon the output. For example, I want to have the molecule with good activity. Now it will see the patterns, give the weightage, which properties makes the molecule more active. Okay, for that it will give more weightage, and based on that it gives the result, desired output as such. Okay, and that is the reason. Okay, in the most of the artificial neural network, okay, it have input, hidden, and output. Okay, so hidden contains the supervised learning methods to generate the the output, the model generation as such. Okay, and yeah, now till now we discussed something like these are the methods, and in the first slide I told that there are n number of data available, and from there we will get few molecules as active and a few molecules as a drugs. If you use the machine learning methods, okay, whatever we discuss, supervised and unsupervised methods, we can channel out some of the molecules as actives from these which are druggable, and then we can complete the pipeline and come up with the drug molecules as such. But the problem here is in the drug test, there is another problem, inherent problem. The available chemical space, whatever I told you, it is 10 to the power of 60. But in the literature, patents and data, whatever you put it, okay, known synthesizable is 10 to the power of 60. Synthesized compounds, whatever you put together, it's something less 10 to the power of 12. Okay, there are several other computational methods we can increase up to 10 to the power of 18. When you use 10 to the power of 12, that means a part of the ocean you are searching and coming with the drug. Okay, if this part you are searching, and especially the drug is in this part, then using the available chemical space, chemical synthesizable space, you can never get a drug molecule. So you need to increase the space such that you can search and find out the new molecule. That's a major problem now. The people are the drug discovery suffering. Okay, because we have a limited space. From there we are searching. Finally, whether we come up with a drug or not is a difficult task. Okay, that's the reason we need to increase the space, and after increasing the space, chemical space, and then we need to search in an intelligent way and come up with a drug molecule. Everything we need to do in one go. Okay, it's a very complex process. Okay, increasing the chemical space, and then we need to identify that predictive molecule, active molecule, and then it should be druggable. Everything we need to have it. Okay, that can be implemented in a method called as a deep chemistry. Okay, deep chem. Okay, in the deep chem, they use a deep learning methods, and then they try to implement the deep learning and try to generate the new druggable candidates as such. This is one of the very buzzing. Now deep chem is a buzzing word in the drug discovery, and there are many companies, okay, who came up very quickly. Okay, they started with a startup and generated billions of molecules by collaborating with the different companies. Okay, so what is the buzzing term now is a deep chem, which address the chemical space. Okay, chemical explosion as such. Okay, so what this deep chem will do, so it's a complete framework. What this do, it takes the database of compounds, it try to understand the patterns. Okay, and the patterns are nothing but the chemical small groups. Okay, now these patterns are nothing that zeros and ones, is a fingerprints. Okay, now what it will do, it try to modify the fingerprint. Okay, and try to generate the new chemical space. Now it will mix and match these fingerprints and generate the new chemical space. Like that, that is nothing but it try to use this deep chem methods. It try to generate the new chemical space, or we'll call it as a latent space. Okay, by using in the deep chem framework as such. Okay, try to identify the patterns and then manipulate the patterns and generate the new chemical space. And then after generating the new chemical space, new molecule, I got a new molecule. If you have a predictive models, okay, or if you have generated a predictive model means the model which have input data and a regression, and we know that these patterns are important for the biological activity. If you have a predictive model, now the new generated chemical space will be passed to the predictive model, and the molecules whichever are having good activity, good druggable properties, whatever, okay, will give it as a final output. Okay, you generate the new chemical space using the predictive models, you try to predict the molecule's property and then generate the new molecule. Okay, sometimes this is called as this is a generative models, these are called as generative methods, and these are the predictive models, and combining together you'll give the drug candidates. Okay, sometimes what these deep chem methods are very dynamic. Okay, what they do, okay, deep chem methods are very dynamic. This is a very important thing, another buzzing word. You try to see the patterns and generate the chemical space with the help of the generative models or predictive models. It will see which patterns are important for the activity, then that information will pass it to the generative model. Okay, now in the generative model, what it will do, it try to generate the molecules, try to generate the molecules which has a desired properties. Okay, it generate the molecules which have a desired properties. That means it need not to pass to that. Okay, it try to use this information and try to generate the new molecules with the properties, like that. So it's a dynamic frameworks. Okay, different frameworks in the deep chem generate the generative models will generate the new molecules, and predictive models will predict and give the drug candidates. Sometimes you use the framework in a dynamic way and generate only the best possible compounds. Okay, like that, it can increase the push up to the chemical space up to 30 to the 10 to the power of 30, or it will wide cover that chemical space, and we can have a high chance of getting the drug candidate for this. Okay, that is the reason this deep chemistry, deep chem, deep learning methods became very, very popular. Okay, we need not to synthesize those many molecules. It gives the desired molecules. It try to generate the chemical space, enumerate that, and then give the best molecules to you. Okay, so that's the reason the drug discovery became very, very highly accelerated, we can say that. Okay, and coming with the molecules. Okay, and few companies, they got the molecules. Okay, maybe within a maybe 6 months to 1 year of time, IND, which went into the phase 1 clinical trials as such. Okay, so that's what deep chem. Okay, just this is the same thing in a other way. So what is this deep learning? This deep chem contains deep learning. What it will do? Okay, simple thing. These deep learning methods are used, especially came up with the concept to identify the human faces or some patterns, etc. Okay, for example, you have given with different what you can say that parts of the human face, and then now slowly you give that that these are the nose, ear, and etc. Okay, so these hidden layers try to identify the pattern, because the output layer, this is what the desired result we want it, and this is what the problem they have given. Now we need to match and make it into this. Okay, for that, in the hidden layers, it will give weightages to each neural network. It is a ear, it is a nose, all these things. Okay, it have a hidden layer patterns. Okay, and then it try to generate different what you can say patterns, and categorize them to ears, nose, etc. And these ears and nose belongs to whom, that it try to categorize and put it into the human faces as such. Okay, as you know, in the simple neural network, it is going to have a input and output layer and with a hidden pattern. Okay, but in the deep learning, it is a connection. It's a network of different hidden layers. Okay, to understand the complex problems. Okay, because it doesn't have only one phase, phase, eyes, etc. Many things are there to understand the complex problems, and to generate the complete image of it. It has to have many hidden layers for it as such. Okay, that's what the architecture of the hidden layer in the deep chem. And these deep chem are now very much used in the in the medical diagnostics as such, image processing, etc. Okay, and this deep chem uses both the what you can say that supervised and unsupervised methods to get into the the image or any kind of processing. Now we use this DNN methods in deep chem to generate the new molecules. Okay, and a small glance. Okay, I don't want to put it because you will come across when you open the deep chem. Okay, you come across this terminology: deep convolution neural network, recurrent convolution neural network, fully connected feedback neural network, deep auto encoder neural network. Okay, and generative adversarial neural network. This is very popular. Okay. Uh, so generative adversarial neural network contains generative and also classify. Generative method means it try to generate the chemical space, and then it will classify based upon the unsupervised methods as such. Okay, like that, there are several types of deep chem methods to address different problems. Okay, just 2-3 more slides. Okay, I will tell you what they will do it. Okay, convolution and other methods, and then we'll jump on to one of the case study as such. Okay, maybe it is little boring. Okay, but yeah, these method, when you open the deep learning and machine learning things, these are the commonly used terminology. Okay, so you should not be after this presentation, you should not be frightened to open and read those things as such. Okay, you can at least this base can helpful for you to understand what they are asking, what they are telling about it as such. Okay, now in the convolution neural network, what they mean, this is mainly used for image classification as such. Okay, for example, okay, these image, okay, of a bird or whatever. Okay, for a number of birds, I want to number of items, I want to identify the bird, and I want to identify the other objects. Okay, so those object, okay, images already feed into the deep chem, and using that, okay, objects and their pattern recognition, it try to identify, okay, in this total images, which is car, which is human face, it try to identify based on the pattern feedback, whatever it got it from the human hidden layers as such. Okay, simple, for example, for a here sparrow. Okay, for each convolution means each curve or whatever, it try to have in the form of tile. Okay, these tiles will have each curve, and these tiles will be used, okay, to generate a to connect to each other to generate a image of a sparrow. And then it says it is a bird or sparrow, whatever it might be. And in the same way, sunset, it may be having the sun with color different, it have it in the form of a tile curve patterns. Okay, and this will convert and make it into a image. and recognize it as a sunset, dog, rat, like that. So for each image, it will have a proper image processing. That's what in the form of a tiles. And then based on this, image processing, it will try to recognize wherever it is any image if you have given, it recognize whether it is a dog or cat or etc. Such. Okay. So this is what the convolution neural network. In remember this, either neural networks or maybe other methods, what regression methods. The thing is, we need to give the input data. Based on that, it will train it and then store it and then recognize the new things. These are the funda for anything. Okay. In the same way, this convolution network for a given molecule, okay, cox3 inhibitor, okay. Now you try to see the image of this molecule, and then in the hidden layers you try to see the patterns how it will be stored, and now based on these patterns, now you try to identify the new molecule which has similar pattern as such. Okay. There is another term called the autocoders. I don't want to bore you, but just two small glimpse of it. Auto encoders, very important. And these are not something like, these are all frameworks which comes from the supervised and supervised learning methods in a proper framework, automated way as such. Okay. Okay. Now in the auto encoders, we have a encoder and decoder. Two things are there clubbed together. What they will do is, it's something like, you try to refine the data. You have a large data. In the large data, okay, you try to take the best data, and then for that data you try to generate a proper model. Okay. To do that, okay, the people use it as an auto encoders as such. Okay. This encoder try to make the large data into very simple way by taking the representation of all the data representation data from the given data, and use it for training the model as such, especially to reduce the noise. And reconstructor, what it will do, whatever it got it the encoder, it try to use that patterns and reconstruct the input data. Okay. That means something like, something like that. Okay. It takes all these data points. What is our thing is, this is the image parts of it. Okay. It takes all the points, and then using autocoders and encoder, not here it is not that autocoder, whatever it might be, but it try to see the patterns and then reconstruct to the image. In the same way, these auto encoders and decoders try to identify the representative data with the encoder, and then with the help of the representations it try to reconstruct the data, okay, given data. With that, based on these two things, okay, try to train the model actually which patterns makes the molecule active and inactive as such. Okay. So there is another important thing is a dynamic model called as a reinforced learning. Okay. What it will do is that thing called as, it's mainly used in, most of the time they use it in the deep chem as such. Okay. Okay. In that, it's a dynamic, actually it's a dynamic learning method where it try to take a state of a molecule and do an action. Based on the action, whether that state, new state, is having a better quality or not. Better quality means is it druggable or not. Okay. If it is a druggable, then it will give a reward. It's non-druggable, okay, it give a penalty. Okay, it give a reward, and no, this molecule is having good reward. Okay. And you need to generate a molecule similar to this molecule. Based on this critic, it take the action and generate a new molecule and will have a better reward, like that. Okay. It try to train by itself, learn by itself, generate the model, I mean by generating the model, and generate the best molecules out of it as such. Okay. So when you have different states of the molecule, okay, it takes that one of that, okay, and add a new fragment and say this fragment is good or not. If it is good, then it takes that action and come up with the next step. Okay. Like that, there are reinforced learning methods. Just skip this. Okay. Maybe much more bored, but all these deep chem methods, in the deep chem, okay, it takes the input data, generate the supervised method. In that, it has a predictive model and generative. It generate the chemical space. Predictive model uses to predict the generated chemical space whether that molecule is active or not. Okay, now in the help of the reinforced learning methods, okay, then it understand which molecules are better, which molecules are bad. Okay, based on this is reinforced learning dynamic learning method, it generates the molecule space with good properties as such. This is a framework for a complete, what you can say, deep chem drug discovery, and this is becoming a very popular, and people, there are many we can say that few companies who are having this kind of frameworks and working on artificial intelligence to come up with a new chemical space addressing different diseases as such. Okay, and there are several validations are there, just everything cannot be covered everything. Okay, like whether the molecule, the model which has generated has a how much false positive rate, true positives, false negative, true negatives. Okay, false positive rate, ROC curve we'll call it as, means the model whatever we have generated is picking the right molecule or not. It is picking the right molecule or wrong molecule. Right molecule is true positive, wrong molecules is false positive. Okay, based on that, okay, what is the precision of that model, okay, how much accurate that model is, that we will predict by using these validations as such. Okay, but yeah, validation is another big thing. Okay, just a small glimpse of it. Okay, and then there are several machine learning tools. Okay, I mean commercially available, and also the I mean publicly non-commercial tools are available. Okay, PyTorch, okay, Skykit-learn, NLKT, Keras, these are all free. SAS, SAS is a commercial tool as such. Okay, NumPy, and these are all free tools available as such. Okay, you can learn these tools and implement this framework for your drug discovery as such. Okay, now one of the case study, we came up with a broad spectrum anti-cancer drug candidates using this AA method as such. Okay, so what we did is, okay, in this just another 2-3 minutes, okay, we took all the anti-cancer compounds from different databases and from the commercial libraries, okay, and processed it, and removed all the peptides or what kind of molecules or maybe proteins, whatever, and took only small molecules compounds. Okay, and then we took those compounds, okay, and made it into a cancer database. Okay, and those are most of them are look like a small molecule database as such. Okay, and these are the patterns come of the similar, I mean common patterns which are there in the those anti-cancer compounds whatever we got it. And these molecules, okay, we took it and clustered them by using this hierarchical clustering. For each cluster we generated a deep learning models. Okay, this deep learning, each deep learning model will have a pattern which says that whether it is anti-cancer or not. Okay, now with these deep learning models we generated the models and screen the, because we didn't generate the new molecule space in this we using this, but we screen the vendor databases whether any vendor means they have ready made available compounds. Okay, so that we can buy, we can skip these synthesis. Okay, we screen the vendor databases and found the molecules which are having anti-cancer activity, and those molecules we tested those compounds, we got active compounds. Okay, and from those active compounds, okay, once again we modify the compounds. Okay, by tweaking the substitutions, etc. And finally we got three compounds which we'll call it as a broad spectrum cancer compound. What is the meaning of broad spectrum? Means these three compounds are active against lung cancer, also active in breast cancer, melanoma, prostate, CNS, renal, leukemia, etc. And they are not active against normal cell lines. Okay, they are non-toxic to the normal cell lines, but active against different cancer. That's what we got as a, because we used the data, all the cancer data with different cancers, and then generated the model. That's the reason we could able to come up with a broad spectrum anti-cancer compounds. And now we can see that all the molecules on the cell line, they are all in nanomolar range activity, these three compounds. And out of that, okay, one of the compound we used for the lung cancer, because we cannot test for we cannot do in vivo studies for all the I mean cancers because it's a money churning actually. So we use a lung cancer, which is because lung cancer is one of the very we cannot say important, but more deadly and most commonly occurring as such. Okay, even for the small non-smoker because of the pollution, etc. That's the reason we use the lung cancer. And for the lung cancer, we have a compound called as BSC16, C16, that is highly active, which is something like, growth inhibition of 40 to 76%. Okay. And the lethal concern means the toxicity is very, very less. Okay. It's more than 100 micromolar activity. And this molecule is very different with respect to the existing drugs. That means that this molecule will do in a different pathway. Okay, and we did the in vitro ADMET studies. Solubility is one issue, we got it, and we did several formulations, and we got some formulation with some clear solution and use it for animal studies for 24 days. Okay, we along with the the vehicle and the erlotinib as a the reference drug, and our molecule we use it. Okay, these are the things which use it for the study. And we see that, okay, our molecule relatively good than the erlotinib. Okay, in the tumor volume reduction or tumor growth inhibition as such. Okay, so this the vehicle is very high as such. Okay, so this indicates that this molecule is comparatively good with erlotinib. But one more thing, this molecule is also active against the erlotinib resistant cell lines. Okay, so this is active against the cancers and also active against the erlotinib resistant cell lines. And you can see the tumor weight, okay, with respect to this molecule have been reduced very great extent when compared to erlotinib and other things. You can see this molecule tumor for the vehicle how much, or molecule how much, and the erlotinib. Okay, and body weight has been much change doesn't happen, not much fluctuation. Okay, even the necrosis, extent of necrosis in the tumors. Okay, necrosis factor, if it is high, that indicates that it is reducing the cancer. That's what one of the indication as such. Necrosis factor is very high when compared to the drug, whatever it is there in that. Okay, so that indicates that this is molecule is very superior. And we did the other plasma availability, the drug availability in the plasma and also in the tumor. And a tumor highly it is in high concentration. And we did other clearance and other studies in the animals, ADMA studies, and they are quite good. And now these molecules, we need to do the IND enabling studies to push into the clinical stage as such. Okay, this is what the stage. And this study we did it with, maybe we can say that, all to put together, we completed this study in vitro study in the 2000. We started in 6 months of time, we completed the in vitro study. And after that, this corona came. We didn't have the animals, and these companies are not available. Once the after the lockdown, we completed this animal studies. Okay, and now we are looking for the other GLP methods as such. Okay, this is what, in a very quick, what I want to tell you, that if you want to have a medicinal chemist, you want to do it, it takes 3 years to 4 years to come to this stage. And maybe whether you can come up with a broad spectrum anti-cancer or not, we don't know. But we could able to do it with a 6 months of time with artificial intelligence protocol as such. Okay, but you can do it any place if you have, because there are many tools open source available. You need to learn but in a proper way, and then you put the framework. Okay, because there are many tools available in this is what I want to stress it. You need not to do much coding. Okay, you need to just have a small knowledge of that, and you can automate it, and you can implement this protocol and come up with the druggable molecules, okay, in your lab. okay, and you can collaborate with the big pharma with your molecules. Okay, that's how you need to do it. Okay, most of the times in India we generate some molecules and we publish it and say we completed our PhD or whatever it might be. But the thing is, if you do it in a more intelligent way by using these rational methods, okay, you may end up with the druggable candidates in your PhD thesis, or maybe you can collaborate with the big pharma to push your molecule to the next stage. Okay, that is a reason. Okay, I want to bring this machine learning and artificial intelligence methods to be rigorously implemented in the labs, in the chemistry or drug discovery labs, to make the molecules more the molecule what you synthesize should be more drug like as such. Okay, this is what about our my presentation. Okay, and my ID if you want to contact me, and we are also open for the to take the interns. Okay, so if anybody want to work with us, you can, yeah, work with us. Oh, sorry. And have any questions? Right.
Thank you, sir. Thank you so very much for your exceptional presentations. It was very remarkable today. And the day you started with the artificial intelligence and taken down to the machine learning and then to the deep learning and started with a beautiful example of case study. Thank you so very much once again for such a remarkable presentation, sir, actually. And please accept your appreciation too in this regard. And if you permit us, we'll go for the Q&A session, sir.
Right. Thank you very much. And yeah, we will go for it.
I request my colleague Sunil to scroll these questions, please. Thank you.
So Balaji has asked me some question about the opportunity to try and get the training in the machine learning. It's our really we want to do a drive actually for the academic people from different academic people to give the training on these methods so that they will be enriched in their lab so that they come with the better molecules from their lab. That is what we want to do it. Definitely we will help it in that. Okay, so definitely Balaji will do that as such, right? Any other?
We'll go for next question.
Where is the, yeah, can the, yeah, definitely, yeah, Sailendra, definitely deep learning methods are useful. Not only the um, type of tumor, okay, and they have deep learning methods, okay, based on the type of a tumor, okay, and they try to identify the what kind of what you can say that what can be the biomarker and all those things they can predict it as such. Okay, definitely there are several examples are there, but we didn't work on that. Okay, but I just gone through several papers where they have done these kind of things as such. Yeah, definitely we can do that.
Thank you, sir.
Yeah, image resolution, because a simple thing, if the image based on the input data, okay, our result will be the precision of the results will be depending on. Okay, so if the image resolution is inconsistent, maybe sometimes difficult how much resolution, it depends on it actually. Okay, yeah, yeah, otherwise borderline, okay, we can say that this can be the bird or something like that, you want to classify it. Okay, we can do it, but the results may not be so accurate as such, right? And depends upon the resolution what we do it, right?
Okay, sir.
Yeah, this is a we can say not $1 million question, it's a big $1 billion question actually. What strategies can be applied if we fail earlier rather than in the late stage of drug discovery? Okay, no, sorry, it's not a $1 billion, because if it is earlier, okay, it's good. Okay, what we do is the drug discovery is not something like it's not a straightforward thing. Always failures will come across, and then we need to optimize our method and we need to go forward as such. Okay, but however, if you have this kind of these tools, okay, engine equipped with you as such, okay, the earlier failure rate we can be reduced. That's what, okay. Earlier failure rate, the most of the times earlier failure rate will be of two things. One is based on efficacy, and another thing is another important thing is solubility or permeability, okay. Okay, solubility comes to the next level as such, okay. So these two things are very important. Okay, when you have a very good in silico strategies, that okay, what kind of molecules we are going to synthesize it, okay, you can use these tools and then identify the better molecules, okay, with have good efficacy, selectivity, etc., and then good permeability. Definitely the molecules definitely can go into the little late stage of the drug discovery as such, okay. So this if you are failing, for example, still with this earlier stage, means your methodology or the tools whatever you are using may not be so accurate in doing it, okay. So you need to optimize the tool once again as such by doing this work, okay, right. Yeah, whatever I just told, NumPy, okay, PyTorch, okay, these tools are all free. In the Python, they have these, okay, DeepChem tools, okay. You can use these tools for your research, but you need to, because all the academic tools may not be well connected to each other, but you need to have little bit more little training to do that, okay. So if definitely if you need those things, okay, we'll help you out on that as such, right.
Thank you, sir. We have another question now.
So internship duration will be 6 months. Say 6 months compulsory, okay, daily based, because something we want to get it some fruitful result, okay. So 6 months is completely required. Cost for sample for cardiotoxicity, okay, we don't do those things actually. We are integrated drug discovery people, where we will do the milestone based research, okay. So for internal and for the clients, okay, not for just for prediction for one thing. We do that. If you want, we can do it friendly based. For example, you want to do it for academics, we'll do it friendly note as such, okay, that you give the sample and we predict it and give it to you as such, okay, right.
Thank you, sir.
Right.
It's over to the QA session, and time to respect your time and honor your chair always. We,
Thank
our
you.
management, the principal, and the college organizing committee is always a pride to have you on this platform, and please accept our honor and
Thank,
in virtually, sir. Please.
thank you. Thank you very much. Thank
Here is
you.
a certificate of your
Thank you. Thank you.
presentation, and you can
Thank you.
receive a memento
Right.
for this too.
Thank you. Thank you. Right. Thank you. Thank you.
Thank
Just
you,
I
sir.
visited your college maybe 2015, I think so.
Oh, nice, sir. Nice to see.
Yeah, I
Nice to.
visited. Yeah. Yeah. Dr. Dr. Abdul Azam.
Yes, sir.
With along with him, I just visited some
Yes.
presentation. Right. Right.
Thank you, sir. Wish to see you and wish to join you in offline as well as online in future more seminars too. It was a truly a remarkable presentation, sir.
Thank you. Thank you very much. Nice.
Thank
Nice.
you so much for
Thanks.
joining. Thank you
Thank you.
so much.
Right. Right.
And dear participants, kindly join us for the afternoon session by 2 o'clock, which
So
is by
I'm
Dr.
signing up. Thank you. Right. Sorry.
Yes, sir. Thank you. Kindly join us by 1:50 p.m. for the second session of today's online eSTTP. The session is by Dr. Chandraya Gadugu, NAIP Assistant Professor, NAIPER, Department of Pharmacology and Toxicology, and the topic is on molecular pharmacological tools. And thank you, and see you by 1:50 p.m. Thank you. Bye.